Hello, everyone. Welcome to the Science of Childhood Cancer lecture for today. Uh, I'm Richard Kowalski, a faculty member in the Department of Structural Biology at St. Jude Children's Research Hospital, and it's a pleasure to introduce to you all Trevor Bivona. Uh, Trevor received his undergraduate degree at Vanderbilt University in Tennessee, as is St. Jude. He earned his PhD and MD degrees at New York University School of Medicine in 2005 or 4 and 5, followed by further clinical training at Brigham and Williams Hospital in Boston, and finally at Memorial Sloan Kettering in New York City. In 2011, he was appointed as an assistant professor at University of California, San Francisco in the Department of Medicine, with joint appointments in other departments as well. He quickly rose through the ranks to full professor currently. Dr. Bivona is a cell and molecular biologist and a board-certified medical oncologist. He leads a basic and translational research program focused on signal transduction and cancer genetics and the molecular basis of tumor initiation and progression. His discoveries have provided insight into the function and regulation of critical cancer-driving proteins, including oncogenic receptor tyrosine kinases and RAS pathway genes. Recently, he and his group members discovered a distinct mode of kinase-mediated RAS signaling in cancer via membraneless cytoplasmic protein granules or condensates. And I'm excited to hear Dr. Bivona's lecture on this subject today. Just a few final points. He is a Chan Zuckerberg BioHub Senior Investigator, is an elected member of the American Society for Clinical Investigation. In the past, he's been a Pew, Searle, as well as Kimmel Scholar, and in the early part of his independent career, he was awarded the NIH Director's New Innovator Award. So, Dr. Bivona, thank you for joining us today. We all look forward to your lecture. I just want to mention to all the members of the audience that if you have questions for Dr. Bivona, please type them in the chat, and we're monitoring that, and time allowing, Trevor will address questions at the end of his lecture.
Richard, thank you very much, and it's a pleasure to be here virtually and to speak on this subject, particularly given the spectacular condensate community at St. Jude. So I'll share my slides here. Okay, well, again, thanks, Richard. And as Richard mentioned, today I'll be speaking about an emerging paradigm, we think, in the cancer field regarding kinase fusions and their ability to self-assemble into membraneless condensates and initiate oncogenic signaling by that mechanism through the RAS pathway and others as well. So I'll just start off with the acknowledgement slide, and this is particularly important since I've never anticipated sort of being in the condensate or space and field, and so I've had to learn from a lot of wonderful collaborators, both here at UCSF and externally as well. In my lab, this work was led by Osman Tapule, who will be soon joining the faculty at Memorial, and was a former postdoc in my group, as well as a former medical student, MD PhD student, I should say, Dana Neal, who's now training in Boston in pediatric medicine, actually. And wonderful other collaborators, Bo Wong, a biophysicist and imaging expert here at UCSF, as well as Jiakun Xu here as well. And Mike Rosen, who I'm sure many of you are familiar with, UT Southwestern, who's been working with us now for the last year and a half to 2 years on in vitro reconstitution of the granules that we'll describe today. So just to emphasize, this has really been an interdisciplinary and collaborative area of investigation in my group. So we're all, just for orientation, we're all well familiar, I think, with this paradigm, the conventional RTK, receptor tyrosine kinase signaling paradigm of lipid membrane-based platforms for initiating signaling, whether it's physiologic or pathologic in the case of mutated oncogenic RTKs like EGFR and others that are prime drivers and targets in many cancers. Classically, of course, signaling downstream of these receptor tyrosine kinases occurs by activation and recruitment of adapter proteins to the plasma membrane inner leaflet that initiates signaling down the RAS-MAP kinase pathway. And canonically, of course, just to remind you, RAS signaling was thought to always require a membrane compartment, at least in mammalian cells. And as many of you are aware, at St. Jude, there's been an emerging paradigm in the cancer field and obviously more traditionally outside of the cancer field for distinct organization of cellular processes through liquid droplets or phase-separated compartments, generally the condensate field. And these are classically membraneless compartments. So in contrast to classic signaling events, they're membrane-based. And of course, examples include stress granules and many others, nucleoli. And classically, again, these these phase-separated compartments serve to concentrate key factors that initiate biological processes, whatever they may be. Other recent examples of note, and I'm sure many of you are familiar with this emerging literature, are the T cell receptor in lipid membrane compartments, as well as our old friend in the cancer field, GRAB2, which can phase separate, as well as transcription factors, nuclear components such as AR, ER, and other fusion oncoproteins as well, such as EWS, FLY1, and others. So again, one of the main principles here, just to reiterate, is that these structures, these membraneless structures formed by higher order protein-protein interactions. I should mention, of course, many of the classic examples here also contain nucleic acid. The ones that I'll describe today, the kinase fusions, do not, to the best of our knowledge and ability to rule that out. These essentially function, again, formed by higher order multivalent interactions, so not dimers or trimers, but very high order protein-protein interactions. That's illustrated here on the upper right. Hopefully you can see my arrow. And again, one of the main functions here is these assemblies then are able to recruit and concentrate effectors such that physiologic or pathologic process is initiated by this effector concentration and engagement. And in this case, today, the function here is oncogenic signaling, activation, as well as amplification of the signal. So how did we get into this in the first place? Well, many years ago, my lab initiated a project on fusions that are important in lung cancer and other. These are kinase fusions that are formed by the anaplastic lymphoma kinase, which undergoes a structural rearrangement in lung cancers and many other cancers as well, where actually the kinase domain here, shown on the right, is fused to a protein called EML4, or a component of an N-terminal fragment of EML4. And this creates an aberrant chimeric fusion oncoprotein, which has at the N-terminus the EML4 protein, again, fused to the kinase domain of ALK. And I should mention that by virtue of the structural rearrangement, actually the native transmembrane or other membrane targeting domains in ALK is absent from the fusion. So there is no transmembrane domain that would otherwise be, of course, in the native RTK, and there's no extracellular domain either. It's just really just the intracellular kinase domain fused to EML4. And I'll come back to that as a theme in general later on. That seems to be a recurrent theme, actually, with many kinase fusions. And so many years ago, a former graduate student in my lab, Goran Jurstanovic, was conducting some more traditional cancer biology studies in my lab on this fusion oncoprotein, and he set out on a project to just understand, what were the signaling pathways that this fusion oncoprotein was activating, and were any of them more important or dominant than others. And he conducted that work and published this paper in Nature Medicine many years ago, where he actually found that the RAS-MAP kinase pathway was the prime pathway that this fusion oncoprotein used to drive signaling. Not to say that others weren't activated to some extent, like PI3 kinase and JAK-STAT, but there was really a dependence on the RAS-MAP kinase pathway for the ability of this fusion oncoprotein to signal oncogenically. And as part of that study, and this was in the published paper, he took a look at localization of the oncoprotein. And again, we were just intrigued because the oncoprotein, the fusion oncoprotein, lacked the transmembrane domain. And what he found was, in fact, that it was not localized to a traditional compartment like the plasma membrane, but actually intracellular puncti. And those are shown here. There's a blow up here at high resolution, and you can see these sort of blobs in the cytoplasm. That was interesting. But what was more interesting, at least to us, was that when he mutated certain domains in this fusion oncoprotein, like a domain in the EMF4 component, the help domain, actually that discrete cytoplasmic punctate localization was abrogated, and that's just shown on the bottom here. And more importantly, again, that correlated with a very substantial decrease in oncogenic signaling. So when the oncoprotein could form these structures in the cytoplasm, whatever they were, we didn't know at the time, there was high levels of RAS-MAP kinase signaling downstream, and mislocalization to what appeared to be the cytoplasm here diffusely diminished that oncogenic signaling. And actually, that was the only aspect of the cell biology of this that was in that published paper, but that essentially initiated this long and winding road over the last 7 years, I guess. And so Osman Tapuli, who I mentioned earlier, picked up the project at that point and really set out to just understand what the identity of that structure was. And our initial hypothesis was that perhaps it was an internal membrane-based structure, an endosome or some internal membrane, lipid membrane-based structure. And so what Osman did was to recapitulate this in patient-derived cells, and as well as in controlled systems where we could express the oncoprotein at moderate levels, just to further validate that these structures were forming. And so here's one example of that. This is a patient-derived lung cancer cell line with an endogenous EMF4 gene rearrangement. And you can see that endogenously here by IF, these structures were present, again, reminiscent of what Gorian had found in the express system. We could control this in a normal bronchial epithelial cell by expressing here, for example, EMF4 ALK at moderate but sufficient concentrations to phase separate. And you can see that the granules are forming here. Or the structures were forming there at the time. Again, we weren't sure they were protein granules. And we essentially set out then on an odyssey of negative data initially, again, testing the hypothesis initially that this was a membrane-based compartment. And so shown here is a subset of those data where we essentially attempted to co-localize the fusion oncoprotein with all known internal membrane structures. Here, either generally by incubating with a generic lipid membrane dye, either labeling the surface or early membrane structures such as early endosomes, or incubating for longer periods of time to allow for internalization, and really no co-localization there. And then a panel, and I'm just showing a subset here, really a panel of markers here that included endosomes, lysosomes, not shown here, mitochondria and others, were all negative essentially for any overlap that we can detect through substantial effort actually. Dana turned to some subcellular fractionation techniques to, again, try to determine whether these were soluble and therefore soluble structures are therefore likely to be lipid membrane associated. And so, for example, she performed classic subcellular fractionation with no detergent or detergent here. And she could solubilize, for example, a lipid membrane-based marker or multiple of them, actually EGFR here, or EEA1, which is an endosomal marker, as you know, calnexin as well, an ER marker, but could not solubilize EMF4 ALK from the pellet into the supernatant, so really no effect. And this was reminiscent of DCP1B, a common biomarker, as many of you are aware. So these were the first clues that perhaps this was not really a membrane-based structure that EMF4 ALK was residing in or on, and so we, through a lot of discussion with folks at UCSF and a little bit of literature searching, we turned to the alternative hypothesis that perhaps these were not lipid membrane-based structures and that they were some form of a biomolecular condensate. And so we began partnering with Bo Wong's group at that point. Bo is a real pioneer in super resolution imaging, and now many generations of advancements beyond that. And so this was many years ago. So this is a snapshot, a picture of an EMF4 ALK puncta again in the endogenous setting by Bo's group, super resolution imaging. And so this does not really look like a lipid membrane-based structure, as you might be aware. This is quite peculiar, actually, even within the condensate space, as far as those in the field who have imaged many of these can tell. It's curvilinear. There's quite a bit of surface area. It's porous in many spaces, and obviously it's quite distinct from the diffuse background of cytoplasm. So it is phase separated. One characteristic of condensates, as many of you know, is that they contain a wide range of sort of liquid to solid-like phases, and I think this is a real emerging area in the field in general. And characterizing this is, of course, of prime importance to understand the biology and underlying mechanisms here. So we conducted fluorescence recovery after photobleaching of the ALK fusion granules here, and this is the most common ALK fusion variant in cancer, variant 1. There are other variants, and I'll come back to that later on, that are less common and actually, we believe, also phase separate, but with some distinct properties, which is quite interesting, at least to us. And so here are the FRAP traces for EML4 ALK variant 1. It's actually quite low level of recovery. So this was an indication to us, and there is some heterogeneity here amongst different granules. But this was an indication to these characteristics coupled were an indication to us that this might be more of a solid-like state for these particular granules. Not totally solid, but more solid-like than more fluid-like. So these were initial exciting clues that perhaps we were onto something here, that this was a membraneless organelle that fit within the general paradigm of a biomolecular condensate. That raised an interesting question. If these were cytoplasmic structures that were membraneless, how could they possibly activate RAS? I mentioned this earlier, and many of you know, canonically, and it's absolutely true that under almost all conditions, RAS requires a lipid membrane-based structure to signal, and that's because, RAS, shown on the right here, but in a nice figure from Mark Phillips, RAS is born in the cytoplasm on free polysomes. It's not a transmembrane protein. It's a peripherally associated membrane protein, GDPase. And the way it associates with membranes is by a series of post-translational modifications, including farnesylation and other modifications that I won't dwell on here, but essentially all RAS isoforms are trafficked by some mechanism to lipid membranes only after this post-translational processing. But essentially, RAS by definition is a cytosolic protein at multiple points in its life cycle, including right after it's synthesized. And so it's certainly possible, and we entertain the hypothesis that perhaps this structure was activating RAS from the cytoplasm. If that were true, we might expect then that certain key adapter proteins that are known to be key intermediates between an activated RTK and RAS might be present at the granules. And so we set out on those experiments initially. So what we're looking at here are localization of EML4 ALK in magenta or purple, and endogenous tagging using it with amion green here of adapter proteins like GRAB2 and SOS and GAB1. SOS, of course, is the classic exchange factor that activates RAS, initiates RAS GTP loading. And we, in fact, and this technique was developed by Bo's group in the BioHub, very powerful, a little bit tricky, but very powerful. To monitor endogenous expression of any protein of interest. And so we actually found that upon expression of the EML4 ALK, these signaling proteins, which otherwise resided in a fairly diffuse localization in the cytoplasm endogenously, as one might expect, were actually quite dynamically recruited to the granules, and excellent co-localization here between the ALK cytoplasmic granules and these different adapter proteins. We validated this at the double endogenous level in many cases. So here's one example where now in the patient-derived cancer line with endogenous EML4 ALK, we also see endogenous recruitment or localization of GRAB2, again, to the cytoplasmic granules. This appears to be an active signaling event by a variety of approaches that we've utilized to show that. One is simply treating with an ALK kinase inhibitor. This was actually the first FDA-approved ALK kinase inhibitor, crizotinib. There are now much more potent ones approved. But in any case, the proof of principle here is shown. So treatment with crizotinib actually caused a dynamic de-recruitment or loss of these adaptive proteins from these ALK fusion cytoplasmic granules. Again, suggesting that the kinase activity was important for dynamically recruiting these, as one might predict from a conventional signaling event of activation of an RTK by phosphorylation of C-terminal tails and docking of these proteins onto those phosphosites. So, is it possible that perhaps, again, RAS was being activated in situ at these granules in the cytoplasm? One initial experiment that we did to get at that, and I'll show you a more direct experiment after this, was to actually utilize a very old, well-characterized mutant in the RAS field that abrogates the ability of RAS proteins after they're synthesized to undergo the post-translational modifications that endow it with the ability to associate peripherally with lipid membranes. And that is a mutation of the so-called CAX-box cysteine at the C terminus. This is the cysteine that is prenylated, farnesylated in most cases, and that initiates the post-translational processing events that again allow RAS proteins to localize to membrane structures and signal productively. So we utilize this mutant or wild type forms, and so when we express, for example, wild type KRAS in cells, either with EMF4-ALK or with control here, oncogenic EGFR, which is plasma membrane localized predominantly. We see, expression of either of these oncoproteins, either the fusion RTK or lipid membrane RTK, can activate wild type KRAS here as measured by RAS GTP levels using a conventional assay. And quite strikingly, and we found that with the SAX mutant here, the C185S mutant that cannot be prenylated and therefore is essentially locked in the cytosol and the cytoplasm, that the mutant EGFR could not activate this cytosolic species of RAS, as one might predict. This is membrane associated, but that the ALK fusion actually could. And so now there is no ability of this to associate with membrane. So although this is a tricked up system because it's a mutant form, this was at least proof of principle that that a cytosolic species could be activated in situ potentially by this fusion RTK in the cytoplasm in the absence of a lipid membrane-based platform. This, again, appeared to be a dynamic process. So if we treat, again, with the ALK inhibitor, crizotinib here, we can decrease levels of GTP-loaded wild-type KRAS, as one might predict, just control here. But same is true for, again, this cytosolic mutant species of KRAS here, the C185S. Again, indicating a dynamic signaling process here in this context. So could we establish more direct evidence for activation of RAS signaling in situ here at these membraneless, what appear to be membraneless protein granules? So we turn to another assay, now conventional assay in the GTPase field, in the RAS field, where the RAS binding domain of RAF1, which is a major, as you know, effector of RAS, this binds to RAS GTP, active RAS, with over 1,000-fold affinity than inactive RAS or RAS GDP. And that's the basis for initiating the RAF-MAP kinase signaling cascade downstream of RAS. This system uses a GFP fused to 3 tandem RBDs of RAF1. And again, is an established cell-based reporter for RAS GTP that essentially reports a spatial readout of RAS, where RAS GTP is in cells. And so the reporter is shown here at baseline. It's expressed diffusely in the cytoplasm and nucleoplasm. There is likely a cryptic GF nuclear localization signal. That's why there's enrichment here in the nucleus. This has been published many times previously. This does not interfere with the ability of this to report RAS GTP levels reliably, and that's shown here. Here's expression of the reporter, now in the context of also co-expression of oncogenic KRAS G12V. So obviously this is a predominantly GTP-loaded form of RAS, and you can see now dramatic localization of the reporter to where you might expect, again, conventional KRAS residing on mostly the plasma membrane here, and you can see the reporter is reporting that. So this was just positive control that the system is working. So where was this reporter localized in the context of the ALK fusion oncoprotein? So again, here's the control condition, and now we find that when we express the fusion oncoprotein here, EMF4-ALK, we now see recruitment and localization enrichment of this RAS GTP reporter, again, to those granules, indicating that RAS GTP was present in situ here and likely being activated in situ at these cytoplasmic membraneless structures. This appeared to be specific, so here are some specificity controls. So if we express a dominant negative form of RAS that interferes with activation of RAS GTP by SOS, by the Grab-SOS complex, and 17 RAS, again, an old classic mutant in the field, we see now loss of recruitment and a mutant form of the RBD reporter that abrogates the ability to bind RAS-GTP, again, a classic published mutant of the RAF1 RBD, again, quite dramatic loss of recruitment of the reporter. So we think this is obviously an active signaling event, not just some sort of non-specific recruitment of the reporter at that locale. And this, we think, is one of the first examples of RAS-GTP localizing to a membranous structure in mammalian cells. So turning back more to the fundamental principles here of the cognate of this particular cognate system and its formation. One hallmark, as I mentioned earlier, is that these are classically higher order protein assemblies, not dimers or trimers or lower order. And so we set out to test whether higher order assembly was critical for the oncogenic output of this by measuring RAS-MAP kinase signaling as one endpoint. And so we conducted structure function studies of this fusion, of this aberrant fusion oncoprotein to try to understand what were the protein domains and determinants of cognate assembly in this case. So we generated a kinase impaired form by making a mutation in the kinase domain, a kinase component of the fusion oncoprotein here. We mutated the help domain. Goring had already done that, but we repeated that experiment. The help domain here in EML4, as well as another domain in EML4 called the trimerization domain, which, as you might imagine, is important for protein-protein interaction and potentially higher order protein interaction. And so here's the native fusion oncoprotein, the quote-unquote wild-type form, localizing to the structures. And all three of these mutants, both the ALK component, the ALK mutant, as well as the EML4 mutants, failed to localize to those structures. So these were expressly diffused in the cytoplasm, as you can see here. And that's just quantified here. Quite dramatic loss of granule formation by each of these mutants. Each of these phenotypes also was recapitulated in the signaling assays. So, for example, EML4 wild type here could recruit GRAB2. I've already shown you evidence of that by IF. Here, this is by co-IP. And either of these mutants, any of these mutants, actually failed to recruit or to co-IP GRAB2. So the loss of localization, again, correlated with loss of the ability to recruit adaptive proteins such as GRAB2. And signaling effects were concomitant. So wild type EML4 ALK here could strongly signal down the RAS-MAP kinase pathway. Here, was measured by phospho-ERK, phospho-MEK. And neither of these mutants could do so. So signaling was strongly abrogated here in the absence of that discrete localization. Again, indicating that the formation of these would appear to be these higher order protein structures was critical for oncogenic signaling. So, and was it really the higher order signaling as opposed to lower or intermediate that was critical? And so for that, to address that question, we turned to a system developed by Jacques Kuhn's lab, Jacques Kuhn-Schuh's lab, very powerful, and there's actually next generation systems that he's developed more recently. This was several years ago. And maybe some of you have employed this. This is the HOTAC system where essentially he generated an elegant approach of fusing any protein of interest. Here, we're going to use the ALK system here as and later other RTK fusions as our proteins of interest to hexameric and tetrameric tags. And essentially, this form allows for very high order protein assemblies and oftentimes phase separation. And so we went back to those mutants that failed to localize or failed to form the cytoplasmic granules. And we engineered them into the HOTAC system. And now, once we did that, we actually, in the case of the kinase impaired here, the trimerization domain mutant and the health domain mutant, now we can recapitulate granule formation, very reminiscent of the endogenous EML4 ALK granules that we observed previously. And these structures were again regained the ability once they were forming to recruit GRAB2, and that's just shown here, at least for the case of the trimerization and help mutants. The kinase impaired form did not recruit GRAB2, even though it formed the structures, and that probably makes sense because this obviously would not have the ability to, and we showed this, it would not have the ability to phosphorylate itself and then to create the phosphodocking site to which the SH2 domain of GRAB2 binds, just like it would to classic RTKs in the plasma membrane like EGFR. Did this forced clustering result in restored signaling? The answer was yes, at least again for the trimerization domain and help domain mutants. So that's quantified here and just shown by Western blot here for ALK and phospho-ERK. So the wild type signals to phospho-ERK. The mutants do not, but when we force cluster them here, the plus signs, at least for the TD mutant and the help delete, we now restore oncogenic signaling here, and not so again for the kinase in point form, as one might predict a bit of a negative control there. So the higher order clustering was actually critical here for restoring oncogenic output, and so we think that's a key principle here, in that the lower order signaling is insufficient for signal amplification, at least at sufficient levels, we think, to initiate strong oncogenic signaling downstream. And actually, I'll come back to that in a few slides. So one aspect of those previous slides and studies that I want to come back to here is that the kinase activity was required for granule formation. So why would that be? So one hypothesis was that perhaps we entertained several hypotheses, but one of interest, and the one that turned out to what we think be true, and I'll show here on this slide, is that perhaps the kinase activity is required, at least in this case, because there are additional protein components that are important for granule assembly, and the phosphorylation of the ALK C-terminal tails might be important for recruiting those proteins. And one of prime interest, given that there's been an implicated role previously for GRAB2 here in multivalency in other systems, earlier published literature in other systems, was obviously GRAB2. So could GRAB2 help explain the kinase activity requirement for ALK fusion assembly, protein granule assembly? And actually turned out to be the case. So I'll just show you here in the red box some IF images that this is a quantification of on the right. So if we knock down GRAB, so the way we went about this was we silenced GRAB2 and simply asked, well, was it required for EML4 granule formation? And so that's shown here, quantified, and here by imaging. So if we knock out GRAB2 in cells that express EML4 ALK, the granules do not form. If we reconstitute with wild-type GRAB2, we can reconstitute granules. And then we conducted a series of, again, structure function studies to mutate different domains in GRAB2, one of which we know is important for multivalency, the SH2 domain. And we also mutated SH3 domains here. And actually turned out that the SH2 domain seems to be critically important for the ability of GRAB2 to encourage granule formation here. And so if we mutate the SH2 domain, we lose the ability to essentially form these granules here. And interestingly enough, that's just quantified here. Interestingly enough, the SH2 domain alone also, when expressed, could partially restore granule formation. So there seems to be, again, a critical role for the SH2 domain, we think, by conferring increased multivalency. And the underlying deeper mechanism is still under investigation. Be happy to discuss that in the questions or in discussion. So just to reiterate, so we think that the kinase domain, or the kinase dependence of the ALK fusion system for granule assembly, is at least partially explained by the importance of the ability of GRAB2 to phosphodock onto the C-terminal phosphorylated tails that are created by the ALK kinase activity. So turning back to this, again, larger question of what is the functional importance of higher order protein assembly, and I think this is a larger question, it seems to us in the condensate field in general, in different condensate systems. And so for this, we actually turn to the different variants of EML4 that are found in human cancers. I mentioned this earlier. Variant 1 is the most common one, and I've shown you only data to this point for variant 1. But what about other ones? There's variant 3, which is present recurrently, but a little less common. And there's a variant 5 that actually is much less common, occurs but relatively infrequently in human cancers. And so why these are interesting is that structurally they're a bit different. So variant 1 I've already reviewed, is the kinase domain here fused to most of EML4. Actually, the variant 3 contains basically the same intracellular kinase domain of ALK, but different parts of, or different structure of EML4 actually only contains a bit of the help domain and the trimerization domain. And variant 5, interestingly, only contains the trimerization domain, so a much smaller piece of the EML4 gene and protein here. And so we just set out to localize these in cells just to see if they behave similarly. And so here's variant 1. I've already shown you this on the left. So it forms granules, as I've already shown you. So does variant 3, pretty similar in general, pretty similar in appearance at least, of these granules. And variant 5 does not. Variant 5 is just diffusely expressed. And so we, at least to the best of our ability, cannot detect any substantial granule formation in cells by variant 5. And that's just quantified here. And what was interesting was that this actually correlated, and I'm not sure this is projecting very well, but this correlated with the oncogenic signaling. So if we look at a number of outputs, RAS GTP levels are shown here, for example, phospho-ERK. If we express variant 1 and variant 3, there's pretty strong oncogenic signaling here as measured by phospho-ERK levels, and much less so for variant 5. And again, this was interesting because it seemed to correlate with the ability or not to be able to assemble these granules in the cytoplasm. Speculating a bit here, but that's also even more interesting because, as I mentioned, variant 5 is much less common in human cancers, whereas variant 1 and 3 is much more common. So perhaps the reason is that this is much more of a weak oncogene. It's certainly an oncogene, but a weaker oncogene that may relate to its inability to form granules versus these stronger oncogenes oncoproteins that form granules. A potential link to higher order and strength of signal and the ability of these to cause human cancers. So what are the biophysical properties of the different granule systems? So I've already shown you variant 1. What about variant 3? Does it look similar? Does it behave similarly? So here's a couple of assays that we conducted again in collaboration with Bo's group. And so here's variant 1, just measuring here now mobile fraction of the granules in cells, in live cells. And so variant 1 is relatively immobile, again, kind of consistent with that more solid-like state that we implicated based on the FRAP tracings earlier. Variant 3 is actually a bit more mobile in cells, interestingly enough. And actually the biophysical properties here are also a bit different. It's actually much better at recovery here in the FRAP tracing. So we think there's less of a solid-like, maybe more of a liquid-like state for these particular granules that may relate, again, to its different structure that occurs as a consequence of the different gene rearrangement that it undergoes. And again, these are definitely ongoing areas of deeper mechanistic investigation as well. So what regulates these granules? I know this is a really interesting and active area for many granule systems. How are they formed? What's their life cycle? How are they turned over? So one area that we addressed at least in the alk fusion granule systems was, are there regulated systems or which regulated systems in cells are employed by these granules or regulate them. And so I'll just show you a snapshot of these data here. So it turns out for the alk fusion granules, they're actually regulated, it seems, by autophagy. And what I mean by that specifically is if we block autophagy here with two chemical inhibitors, chloroquine or baflamycin, we actually impair increase the number and the size of granules in these cells, and that's shown here. And so this indicated to us, at least, that autophagy was helping to turn these over and degrade them by some mechanisms, which we're trying to understand currently. And that was interesting also from a signaling level. In that what we found was that when we block autophagy, again here using chloroquine or baflamycin, there were more granules, the granules were larger, and most interesting, at least to us, was that there was increased signaling. So the larger and the more granules there were in these cells as a consequence of blocking autophagy, there were higher levels of phospho-ALK and downstream phospho-ERK signaling. And this was actually a critical piece of data, not only to understand what systems were regulating these granules and their life cycle potentially, but also to really implicate the ability of these to form granules for their active signaling role. These were not dead aggregates. In fact, if we increased their size and number, they were even more active from a signaling standpoint. So just want to turn in the next few slides to the question of, could this be more generalizable? I mean, EMFR-ALK is an important oncogene, but it's not the only RTK fusion, and could this be even a larger paradigm amongst RTK fusions? And so one experiment we did it was an engineered proof of principle experiment here, where we simply took EGFR, canonical EGFR, and truncated it so that it just contained the intracellular kinase domain, no transmembrane domain, no extracellular domain. And we used that hot tag system again to force to see if we could force cluster it. And so here's the intracellular domain of the kinase domain of EGFR expressed diffusely here, no hot tag, diffusely in the cytoplasm and a bit in the nucleoplasm as well. And then if we use the hot tag system, we actually could force cluster this. And more interestingly, we could recruit GRAB2 to these structures. These were membranous. I'm not showing you those data here, but these were also membranous structures in the cytoplasm, reminiscent of the ALK fusion granules. And these could signal. So if we looked at phospho-ERK levels here, when we force cluster, these actually significantly augmented their ability to not only recruit GRAB2, but now signal down the ERK pathway as measured by phospho-ERK. Negative control, this was not true for the same intracellular domain that we could either force cluster or not with the hot tag system, but that had a kinase impaired mutation, a kinase domain mutation that rendered the kinase impaired here, K78A. So this again was an active signaling event requiring the kinase activity here of EGFR, presumably because it creates phosphodocking sites again to recruit GRAB2 and initiate oncogenic signaling here or proliferative signaling here down the MAP kinase pathway. And I should mention, and this is definitely a genetically tricked up system, but as many of you know, there is a literature from Graham Carpenter and others on truncation or cleavage of membrane-based RTKs like EGFR and potential intracellular signaling roles for the truncated kinase domain. Although, we have no idea whether there's whether there's a role for any condensate function or formation by those endogenous forms. So perhaps a more interesting generality series of studies here, and just to introduce this. So many RTK fusions, not only ALK fusions, but many others that occur recurrently in cancer. So RET fusions contain this common principle that I mentioned very early on, in that they contain the kinase domain of whatever kinase, ALK or RET or others. And they're most commonly, not always, but most commonly fused to an N-terminal partner that contains multi-merization domains, and the structural gene rearrangement that fuses those two genes and proteins actually removes or eliminates the transmembrane domain or obvious membrane targeting domains. And so here's the ALK system. So these are all found in human cancers to varying degrees. Here are the different flavors of the N-terminal proteins. So many of them contain, so there's a variety of them. So EML4 is one that we have already walked through. TFG, RANBP2, KIF5B, etc. And these, again, very commonly contain different flavors of oligomerization or multimerization domains. So coil domains or fibronectin or WD repeats, TPR domains, etc. Same is true for the RET fusions. So many of these, again, different flavors of 5' partners, but many of these contain just the kinase domain and then fuse to whatever N-terminal partner that contains as a common theme. Again, these PPI, protein-protein interaction domains, such as here with KIF5B, the coil domain. One that I'm going to show you studies on in the next few slides is a RET fusion that's important in lung cancer and others, fused to the N-terminal partner CCD-C6, forming the CCD-C6 RET fusion. So is it possible that many of these may form this cytoplasmic membranous structure as well? And again, here's that general theme that we embarked upon testing the hypothesis around. And I should mention, I'm not going to show you data for this, but some ongoing work in the lab now is that this is also true. This theme is also true for non-RTK fusions. So many of them also have this same fundamental principle, at least the structural principle. Whether they form condensate systems is an area of active investigation. So does this fusion of RET to CCD-C6 operate in the same way that ALK does? Well, the answer turned out to be very similar. So here's CCD-C6 RET localized in cells, so you can see it forms these punctae. I'm going to walk through these studies a bit more quickly so that we can allow time for discussions and questions. So this forms granules. These recruit GRAB2, again, reminiscent of the ALK fusion system. And these also locally enrich RAS GTP, again, similar to the ALK fusion system. We conducted similar structure function studies, mutating, for example, making a kinase impaired version of the fusion here, or mutating the coil domain in the N-terminal partner, CCD-C6 RET, again, to test, again, the structural determinants of granule formation and downstream signaling. And we found that indeed the coil domain here was important for granule formation and oncogenic signaling. I'm showing you the signaling aspect here using RAS GTP levels as well as phospho-ERK. and RET assays. The kinase impaired form of RET here, the K147M, diminished phosphorylation of RET, as you might predict, and also downstream signaling. Interestingly enough, I'll show you these data in a minute, but the kinase impaired form of RET actually was capable of forming granules. So one distinction from the ALK fusion system, where there the ALK kinase activity was required here, although the kinase activity is required for signaling, it was dispensable for formation of the RET cytoplasmic granules. And so this is just shown here. What we conducted here was actually, this is a bit of a different experiment. This is actually addressing whether if one fuses the CCDC6 N-terminal partner to ALK, which required again the kinase activity and made a kinase impaired form, could that form granules? And actually, it can. So there were determinants here in the CCD6 component of the RET fusion that were actually sufficient for granule formation, even in the absence of a kinase activity. And so that was a clue that perhaps there were different structural rules between different RTK fusion systems that govern the ability to form granules, and I'll come back to that. And again, we can conduct also the converse experiments here. So now if we take EML4, the N-terminal partner of ALK, and fuse it to RET here on the bottom, and kinase impair it with a mutation, now we lose the ability to form granules here. So there's something about, and I'll come back to this in a minute, we think we understand a bit about what it is. There's something about the N-terminal partner that either is sufficient or insufficient for granule assembly, in which case or not, you also require the kinase activity component. And again, the signaling aspects were similar in that condensate formation was required for downstream oncogenic signaling. So here's our working model that we're continuing to investigate. So we think there's at least two classes of RTK fusion granules, one of which is kinase dependent, requiring the activity of the fused kinase in the case of ALK, one of which is kinase independent, in which case the kinase activity of the fused kinase, for example, RET, is dispensable. And we think the underlying mechanistic explanation is that the kinase activity is required in cases where the N-terminal partner contains domains that are insufficient to meet the threshold for condensate formation. And therefore, you require phosphorylation and recruitment of other proteins, for example, one of which is GRAB2, which I showed you earlier for the RTK fusion system. For the kinase independent class, these fusions actually contain N-terminal protein partners that are fused to the downstream kinase here that are sufficient for valency to allow for the higher order protein assembly here, and therefore this class is kinase independent, and the kinase activity and recruitment of additional proteins is by and large, at least for what we've tested so far, independent, and therefore does not require phosphorylation and recruitment of proteins like GRAB2, such as the ALK system does. So I'll just close out here in the last minute or two. And so classically, of course, lipid membrane signaling is very important for signaling by physiologic and pathologic RTKs. And so, our work in no way diminishes the importance of classic, lipid membrane-based structures, but we think that we may be onto a new paradigm here in cancer biology, where some and maybe many of these RTK fusions and maybe even some non-kinase fusions actually function in this non-canonical condensate-based mechanism, where they self-assemble into membraneless cytoplasmic protein granules. These are peculiar structures. They phase separate, they phosphorylate themselves, and then initiate engagement of downstream effector cascades, such as recruitment of GRAB, SOS, and other adapters here, SHIC as well, and others. And this initiates, we think, in situ RAS-GTP activation and downstream MAP kinase signaling. And I should mention that these condensates also initiate activation of other signaling cascades, but for sake of time, I won't get into that in detail right now. So what are ongoing directions? Well, we're continuing to try to understand the greater protein architecture of the oncogenic protein granules. So we have a limited set of data on which proteins are recruited or not to the granules, but we're conducting more unbiased approaches to try to capture the proteome of these different granule systems. With Mike Rosen's group, I alluded to this earlier, we're trying to understand the in vitro phase separation properties. And so actually Mike's group and data that I won't show today I'll just mention briefly, have been able to reconstitute much of this in vitro using purified proteins, which is quite exciting, and conduct phase diagrams and other more rigorous approaches to understand, again, the biophysics here. We're very interested in, from kind of a classic signaling standpoint and cell biology standpoint, what are the RAS and RAF engagements here? Is there isoform specificity? As you know, there are different isoforms of RAS, different isoforms of RAF. Are all of them participating or some not, in this non-canonical signaling process? Are there differences in proteomes or engagement of isoforms or signaling pathways by different oncogenic RTK fusions? We have a clue that there's some extent of this already by comparing, again, our initial proteomic studies of RTK of ALK fusions with RET fusions. There's quite a bit of overlap in at least a limited proteome we've interrogated to date, but there are also some differences, and so there may be important biology there to understand. What are the downstream signaling pathways? And we focused mostly on the RAS-MAP kinase pathway for many reasons in our initial studies. But we do have evidence that, at least for the ALK fusions and to some extent the RET fusions, they can activate other pathways such as PI3 kinase and JAK-STAT. And how do they do so? Particularly for this one, the PI3 kinase pathway, which, as you know, requires a lipid membrane structure. So we think there's an opportunity to understand some compartment crosstalk here, and how are these membraneless structures interacting with membrane-based signaling events. And then one kind of translational area that we've thought a lot about and are thinking hard about experimentally tackling is, can we leverage this mechanism to develop different or differentiated suite of therapies that don't interfere necessarily with the ATP with the kinase activity in an ATP competitive way, which is what current approved inhibitors of RTK fusions do. But actually leverage the ability to inhibit protein-protein interactions and disrupt granule assembly or effector engagement, again, as a distinct approach to disable this oncogenic signaling platform and therefore downstream oncogenic signaling. I think with that, I'll conclude and be happy to, I think there are some questions that I saw coming up, be happy to enter into discussion and questions. Thank you very much.
Thank you, Trevor. That was really spectacular, very clear, and very mechanistically informed. So yes, there are a number of questions in the chat, and others can add to those. Now I'll read them in order. So from Taosheng Chen here at St. Jude, he asks, can the cytoplasmic clusters, droplets, translocate across the nuclear membrane into the nucleus? Do you ever see them in the nucleus?
Oh, that's a good question. No, I don't think we have actually. I'm not sure we've rigorously tested that, but no, I think in our imaging experiments we've seen them only in the cytoplasm. I don't think we have any evidence to date, at least the ones we tested, that they are in the nucleus.
Yeah, they're probably too big. Okay, Nicole asks, since EML-ALK fusion variant 5 does not form puncta, do you think it uses different mechanisms to drive oncogenesis? And if so, do you have any idea what those mechanisms might be, or does this suggest that puncta formation is not required for FO-driven oncogenesis in the case of that fusion? Yeah, that's a great question.
Yeah, thank you for the question. So I don't have the direct answer experimentally. So we know that it activates the RAS-MAP kinase signaling to some degree. So our working hypothesis for that is that it probably barely meets the threshold for an oncogene, and I would say that we have no evidence for any sort of cluster formation for that one. So I would say that in that case it's independent. It's a cluster content independent mechanism. Like many, our claim is not that all RTK fusions will do this, but that's an example of one that we think does not.
Yeah, so if I could just add to that question. So if I recall correctly, variant 5 exhibited high phospho-ALK, but as you noted, phospho was ERK was lower than variants 1 and 2. Could
Yeah.
you comment on that? That's puzzling.
Yeah, it's a good question. I can speculate a bit. So, I think our what we would propose is that um, there may not be enough effector concentration to initiate the MAP kinase, cascade. And I'm extrapolating from our work on variant 1 and 3, where we did the kind of structural tiling. And so, we could see a clear quantitative difference as we picked off different domains that are required for protein assembly of variant 1 and 3. And when we did so, we kept diminishing recruitment of GRAB2 and phospho-ERK downstream. So I suspect for variant 5, there's just not that local enrichment of enough to get signal amplification to really boost the phospho-ERK signal all the way back up to where you would see with variant 1 and 3. But again, that's extrapolation.
Yep, thank you. Nicole also asks, have you done IP mass spec or other protein-protein interaction studies to identify other proteins that interact with these condensate-forming fusions?
Yes, thank you for the question. Yeah, so we did, but we are now revisiting it. So I'll tell you what happened. So in our initial studies, we of course went after this with a lot of vigor, and we did conventional IP mass spec with it. Nevin Krogan's group here, who's mass spec everything in the world practically, and what we pulled out in terms of high confidence hits were like two proteins, GRAB2 and I think SHIC, and so which we already knew by co-IP. So we've had to turn to kind of more sophisticated systems. So right now we're actually collaborating with a couple of labs doing some proximity labeling approaches, so bio ID and mini turbo ID, to try to more reliably capture the systems. And my understanding, and I'm not an expert in this particular area, but my understanding is it's been quite difficult in the concept field to really conduct at least conventional proteomic studies.
Great, thank you. And Thomas Capito, also from St. Jude, asked a question I think that you already addressed regarding variant 5. So a question that I had was, and I guess this pertains to the fusions that require phosphorylation for condensate formation.
Mhm.
And so do you understand the order of events of condensate formation? In other words, are there protein-protein interactions that cause condensate formation, which is then strengthened by phosphorylation events?
Yeah.
Or is it the other way around? And have you begun to sort of address those sorts of detailed mechanistic questions?
Yeah, thank you, Red. That's a great question. So, I'll say so. We are trying to address those deeper mechanistic questions, and we've done so to some extent. And actually, the most traction we've gotten, we think, is with Mike. So he's been able to start to put together order events, at least in the in vitro system, and then we're going to go back into the cells to kind of test that as well. And so we know phosphorylation is critical, so we think that's probably an initiating event. But just like in cells, in the test tube using purified proteins, Mike's group has shown that you also need GRAB2, and you also one thing I didn't mention is that you also need SHIC. And actually in cells and I think in the in vitro purified protein system that Mike has, SHIC is even more important than GRAB2, it seems. So,
I
so
see.
our working hypothesis is that we think protein-protein assembly by the fusion comes first, followed quickly by phosphorylation, and then increased multivalency by recruitment of GRAB2 and SHIC. Yeah.
Great. Yeah. Yeah. Uh, that's what I would have guessed. So let me probe you about your thinking regarding therapeutics. So, you know, how are you approaching that if you are actually initiating studies in that direction? I guess I'll preface that by saying, I'm sure you're aware of Rick Young's work where they've shown that certain types of small
Yeah.
molecules can preferentially partition into condensates. And so, one can think of, designing molecules that have a component that directs condensate partitioning and then another, moiety that, is the warhead that targets the kinase or a protein-protein interaction. What are your thoughts in this space?
Yeah, I would say this, very fluid at this point. But we're thinking about it in different ways. So one is, one direction that you mentioned. Another is, and Rick's work is super interesting. Oh, just a tangent there, a corollary there. One other aspect that we're interested in is could the condensate, the ability to form condensates, or the different maturation process that these condensates are undergoing actually affect even current ALK inhibitor or RET inhibitor partitioning? And I don't think anybody's looked at that. We'd like to some extent. And that could certainly be clinically relevant, we think. But for the next generation of therapeutic approaches, yeah, I mean, we're thinking about different things. So one is, could we identify protein-protein interaction inhibitors that, for example, interfere with recruitment of Schick or GRB2 to the granules, which we've shown genetically, if you block that, you block granule formation and downstream signaling. So that's one. That's a challenge because traditionally at least, although this is changing a bit, PPI inhibitors have been challenging, but there could be renewed, I think, enthusiasm for that based on some recent chemistry that's come out. And then the other is, one approach actually that we've been thinking about is, could there be a molecular glue approach to some extent where you recruit in sort of an inactivator of something, whether it's phosphorylation or maybe some modulator of autophagy, at least for the ALK fusion system. And so those are kind of the general areas that we're approaching this through. We think that whatever campaigns we conduct, it's pretty important. I'd love to hear your thoughts on this, actually, to conduct cell-based assays. We think that the properties in cells are, going to be probably, pretty specific, to these granular systems. So I know many folks in kind of the PPI space have a lot of times done sort of, purified protein and based, chemical screens, for example. But, we're trying to think cleverly about how to conduct sort of cell-based assays around these different therapeutic approaches.
Yeah, so here at St. Jude, I'm sure you're aware, we're interested in nuclear fusion oncoprotein driven
Yeah.
condensates, and we do have a drug discovery program associated with that work, and we're very much focused on cell-based imaging assays of the effects of compounds
Mhm.
because, for example, in the case of the NUP98 fusion oncoproteins, we know that if you can displace them from chromatin,
Mhm.
they still form condensates, but the condensates are large and small in number, and they're not oncogenic at all. And so if we can identify small molecules that shift the condensates from being loaded on chromatin to being off chromatin, they would probably be efficacious in
Mhm.
down regulating the aberrant transcriptional program that the fusion drives. So yeah, I do think that cell-based assays are the way to go. Although, there's a school of thought though that if you can identify the minimal components of a condensate and reconstitute a sort of skeletal condensate in vitro, that you can recapitulate a lot of the key features of the physical features of those condensates. And so, maybe a parallel approach is maybe really the way to go with
Mhm.
screening done in cells and then more detailed mechanistic study of lead compounds in vitro condensates. So we're now past the top of the hour, and I know, Trevor, that you have a busy day ahead of you. So maybe it falls to me to thank you one more time for a very stimulating lecture. Thank you very much for joining us today, and I look forward to seeing you sometime out in the real world as as
Yes.
conditions allow.
Thank you, Richard.
And yeah, thanks again, and have a good rest of the day. And thank you everyone for joining us today.
Thank you all very much.
Okay,
Take care.
bye now.
Bye-bye.
You too.